What are biomarkers for early Huntington's disease detection? Imagine catching a disease before it even shows its first signs. That's what scientists aim for with biomarkers for early Huntington's disease detection. These are measurable signs in the body that can tell us if the disease is present or starting to develop, even when someone feels perfectly fine. They can be found in blood, cerebrospinal fluid, or through brain scans. This makes early detection possible and helps doctors monitor the disease more closely. One key biomarker is neurofilament light chain, a protein released when nerve cells get damaged. Elevated levels of this protein show up in blood and cerebrospinal fluid long before symptoms start. The higher the levels, the more likely the disease is progressing. It also relates to the number of CAGs repeats in the H gene, which is a genetic marker for the disease. This makes neurofilament light chain a sensitive indicator of early nerve damage in Huntington's disease. Another important biomarker is mutant Huntington protein. This is the abnormal form of the protein caused by the genetic mutation that leads to Huntington's disease. Scientists can detect low levels of this protein in cerebrospinal fluid and blood. As the disease advances, levels of mutant Huntington tend to increase, especially when motor or cognitive problems begin to show. Brain imaging is also a powerful tool for early detection. Structural magnetic resonance imaging, or MRI, can reveal early changes in the brain such as shrinking of the striatum, a region heavily affected in Huntington's disease. These changes can be seen even before movement problems appear. Diffusion MRI can spot microstructural changes in white matter, like decreased fractional anisotropy, which can be noticed years before symptoms show. Advanced imaging techniques are being studied to detect subtle changes in brain function that might signal early disease. Scientists are also exploring inflammatory markers. Molecules like interleukin-8 and tumor necrosis factor alpha are immune system signals that can be altered in early Huntington's disease. These changes might reflect inflammation in the brain, which could contribute to disease progression. Other fluid biomarkers include mitochondrial DNA levels in blood plasma and substances like pronkephalin. These markers are linked to early nerve cell changes and might help predict when brain cells are being lost. Sometimes, subtle clinical signs can hint at early Huntington's disease. Minor motor issues like slight involuntary movements or slowed movements can be early clues before a full diagnosis. Using all these biomarkers together can help identify people carrying the H gene who are at risk of developing symptoms. This allows for earlier monitoring and potential treatments. It also helps track how the disease progresses in people who are already showing early signs. Combining blood tests, brain scans, and clinical assessments gives a clearer picture of the disease at its earliest stages. This approach supports better management and advances in research for hereditary health conditions like Huntington's disease.